I'm Stephen Glazier, Director of Technology at Battery Technology Solution, Novonix division in Halifax, Nova Scotia in Canada. So the overview of my talk, I'll give a quick introduction to Novonix, briefly what we do, and then talk a lot about our UHPC, Ultra-High Precision Coulometry. During that background, I'll give a little bit of theory of what are we really measuring when we're testing cells with really ultra-high precision, and then how can we use it? And I'll actually get into some real-life case studies that we've done in-house, accelerating our own R&D in our materials divisions, and also with some partners. So the Novonix group is a series of small groups, some bigger than others, like our anode materials division, which a lot of folks are really familiar with now. We're commercializing, mass producing our synthetic graphite with offtake agreements with tier ones like Panasonic, PowerCo, and Stellantis. Our cathode materials division, which is also in Halifax, Nova Scotia, where we're commercializing patented all dry, zero waste cathode synthesis technology. And we're looking to license and partner on this technology rather than build it up ourselves. And then also in Halifax is our battery technology solutions division, where we provide UHPC, this cell testing equipment. We also offer R&D services with our pilot prototyping lines and over 3,000 channels of test equipment. So we work with folks all across the industry to scale up their technologies, prove it out, and we utilize our ultra-high precision coulometry in everything that we do. So we make and manufacture this equipment and also are the biggest users of it. So what is UHPC? It is the highest current and voltage accuracy and precision equipment for battery cell testing. No matter what everybody else's advertisements say, they're the industry leading accuracy and precision. We do have the highest precision and accuracy for voltage and current. So one thing that we've noticed over the years is a lot of people will get it. They'll read a lot of papers and understand, a couple of use cases, but then say, like, why should I care? How can I really use this to solve my problems? And that's what we've really kind of set out to solve and to, come to these presentations, come to these conferences and present and try to show real life data because we can't show a lot of our customers' data. So try to find some things that we do internally or even do some of our own case studies to showcase how you can use UHPC. So, what can you do with it? What we really like to focus on is, finding the gaps between what normal cycling equipment can do, and then try to apply that to UHPC. So existing cell test equipment really excels at things where bulk metrics and small uncertainties in raw data don't really matter a lot. So this can be testing cells to end of life, testing cells for hundreds or thousands of cycles, rate capability where uncertainties of 0.1% or so don't really matter. DCIR, GITT, application drive cycle simulation. But really the gaps exist in understanding impacts on lifetime really quickly, trying to do really high throughput measurements, and then test things in weeks or days instead of years. And I'll show some applications that we've done internally that really show how to do this. So this can be used in material and technology development, really just applying for high throughput studies, and then you throw the winners on your traditional cycling equipment. And where we're really trying to work with some of our customers and partners on are using this in QA and QC, really for moving into manufacturing, how you can use this ultra high precision to detect things earlier that could matter a lot later. So we always say we can do anything that a normal cycler can do, but it's really being designed to measure these small differences that matter. So to talk about this, I'm going to go back to the basics a little and then ramp up into like what are we really measuring? What happens within cells and why does that matter? So the only thing we're really directly measuring when we're testing a cell, we're used to looking at this diagram here, charging a cell, counting the number of electrons through there based on the amount of lithium we're moving across from the cathode to the anode. But really, this isn't the only thing that's contributing to electrons moving through this external circuit. It's not just a direct measurement of the lithium moving across the cell. So the thing that we're directly measuring is the potential difference across the electrodes and the number of electrons through the external circuit. And we infer what's happening inside the cell based on that. And that's really important when we're trying to figure out what we can do with this high precision data. So in the following diagrams, I'm just going to simplify things and just show what happens during charge. Because when we throw all these mechanisms together, it gets quite busy. So we're all really familiar with capacity loss. We plot that all the time. As we're charging our cell, we lose some lithium to SEI growth on the anode. We have some particle cracking or electrocracking. We have mass loss happening. So source of capacity loss can be SEI growth, active material loss. Even impedance growth can then affect how much capacity you are able to obtain each cycle. And this, of course, leads to lost capacity, impedance growth over time. And what this eventually does is I'm going to build up this description of coulombic efficiency here. What this does between the charge and discharge cycle, losing a little bit of capacity. So your discharge capacity is slightly smaller than your charge. So you get this ideal coulombic efficiency of, say, 1, and then subtracting this fade term of all of this capacity that we're losing. We're all very familiar with this. Then the question is, what else is happening? What else really matters for degradation mechanisms in lithium-ion cells? So other sources of electrons are all kinds of other electron donating reactions or mechanisms. This can be electrolyte oxidation, shuttle mechanisms. So I just walked through a few of them here. When we have something like manganese dissolution happen from our cathode, in order to create charge balance in our cell while we're charging, we're also going to send a couple electrons through this external circuit and count those. If we have a short circuit, small self-discharge within our cell, we just create this electron shuttle around, and we can be counting that. We can have reversible redox reactions. Great paper by Jeff Dunn's lab looking at PET tape and shuttle mechanisms that happen there. They actually detected these using UHPC. And then irreversible electrolyte oxidation. And so as we're charging our cell, we're counting all of those electrons. We also have all this other contribution if we can measure it. And that's what really UHPC is here to do, is help identify these things that are happening typically below the noise value in other cycling equipment. And so all of this during charge, we're counting all this extra capacity. During discharge, all of these mechanisms act like self-discharge, basically. So we're counting all this extra charge capacity. We lose some discharge capacity into these invisible self-discharge mechanisms, and that also decreases our coulombic efficiency. So now we've got these kind of two separate terms to coulombic efficiency that if we can really measure what's going on in our cell accurately enough, we can start to split up some of these families of different degradation mechanisms. And one really important thing to highlight on this slide is within the very simple boundaries of each of these mechanisms shown, there's no direct loss of lithium inventory. I'm not showing any capacity fade in any of these mechanisms. They may trigger capacity fade later, but within these slippage-based mechanisms, there's no lost capacity. So you can have a cell with lots of these mechanisms happening, and eventually these may lead to salt depletion, impedance growth, and sometimes this is the main contributor to your kneeing, your rollover failure. And so if we can measure these types of mechanisms, if we can quantify this, it can be really useful for doing really rapid development, identifying things really early that are going to matter way out in time that we can't actually see with traditional metrics like capacity fade. So just to highlight what UHPC is capable of, if we line this up with a bunch of other types of cycler equipment that's available and is probably in most labs that people own here, and you throw 8 or 9 identical tier 1 cells on those pieces of equipment and cycle them, you can see these spreads of some of these pieces of equipment. And the coulombic efficiency measurements on NOVONIX UHPC, the spread is 2 orders of magnitude better than any of these other pieces of equipment. So really we're identifying the actual difference between the cells at this point. which, if you want to change your chemistry, you want to change an electrolyte, a cathode material, a dopant, we can start to see very accurately, very consistently, repeatably, differences between chemistries that you may not be able to see in just a short number of cycles on other pieces of equipment. And this was the real motivation behind UHPC, something like 15 years ago in Jeff Don's lab. So the way we do this is obviously just having higher precision and higher accuracy current and voltage measurements. So you can visualize this a lot better just by looking at current, looking at these other cyclers that we saw, and seeing, cycler 1 has pretty good precision but poor accuracy. Cycler 2, you can see the bit switching here, 16-bit resolution. Same with cycler 3, kind of all over the place. And then our equipment. And so what this turns into, when we take all this raw data, we integrate it, we start integrating all of those uncertainties, maybe those biases between charge and discharge. So we're consistently maybe overcounting or undercounting. We see that turn up in coulombic efficiency. These are 8 or 9 identical cells. And so we see pretty good precision and resolution, but very poor accuracy between all of these cells. And then these other 2 cyclers, not so great. With capacity fade, we also see generally this trend, even if you're kind of going up and down cycle to cycle, you get your general trend. This is a cumulative effect. So, you can do capacity-based measurements and look at fade on any type of equipment. And really what makes things stand out is this metric called charge endpoint capacity slippage, which I'll describe quickly in this slide. And this is how we start to quantify some of these slippage-based mechanisms. So I was discussing with those slippage-based mechanisms, we're counting all these extra electrons during charge. During discharge, we're not counting them because it basically just acts as an internal self-discharge mechanism. So what that means is every time we charge up our cell, we're going to go a little bit maybe past our theoretical endpoint or capacity. Then when we discharge our cell, we're going to fall a little bit short. And we're going to do this very consistently cycle to cycle. What that means the next time we charge up, if we start back at where we landed, not quite at 0, we actually typically overshoot the endpoint, the last charge endpoint, because we're going to overcount again. And then we're going to discharge and fall a little short, charge up again. And what this means is our voltage curve ends up kind of walking out to the higher capacities here. And the rate at which it does this relative to other cells has to do with these slippage-based mechanisms, this overcounting during charge, undercounting during discharge. And if we just put a pin in the map at the end of the first cycle there, at the very top of charge, and then track this endpoint as we cycle, sorry, my hand's a bit shaky there, then you can see how this endpoint kind of moves, and that this is very consistent between cells when you have the accuracy and precision to do it properly. So there are a pair of cells here, different types of additives. Additive 1 is just vinylene carbonate in this cell. Additive 2 is vinylene carbonate and propane saltone. And that propane saltone additive really lowers the rate of these slippage-based mechanisms happening at the cathode. And we can see that in a couple cycles. We don't need to cycle these cells out hundreds of cycles to figure this out. We can immediately make a decision. And then if you look at this, the rate of this, basically the derivative of this curve, we can actually say like this is quantifying the relative rate of these reactions happening with respect to one another. And if we look at what this looks like with identical cells on these different systems, you can see that these biases of these, the accuracy of counting between charge and discharge are really poor. So both the accuracy and precision are very important. We can really only get this type of measurement using UHPC. So I went through a lot of diagrams there and some background, and now I'm going to get into some case studies about why this is really useful, how we've used it, and how some of our customers are using it. So there's all sorts of opportunities for using UHPC, for rapid in-house development, short-term cycling to do really high throughput without having to just wait forever for cells to cycle. You can utilize new metrics to do this. For QC, we're looking at using inter and intra-batch statistics, really short-term measurements to determine how different batches or lots of materials are, or cells coming off of a line, or look for variation within those batches. And then, of course, a ton more things that we can do. And I'm actually going to talk a little bit about lifetime prediction using some of these new metrics at the end of my talk. So this first case study is talking about cathode material development. So we are a cathode materials company, so we use UHPC with every single material that we produce. We're really rapidly able to take those materials, move them into our pilot line, build full cells out of them. And then we do traditional cycling because that's what our customers want to see. But before we do that, we do UHPC cycling. And so these experiments here are showing two different cathode materials. These are both NMC 622s. And we cycle these for a few thousand cycles, and it takes 8 or 9 months or so to get to 2,000 cycles. And there's no sign of any difference between these materials in this long-term 1C cycling. Even when we look at delta V here, we're defining delta V as the difference between the average charge and discharge voltage. So kind of a proxy for the DCIR in the cell. There's also no real warning that this is going to happen. And all of a sudden at 2,000 cycles, we have this rollover failure happening. And so the hypothesis here is that UHPC should be able to predict this if it's going to be really useful for our cathode team. So when we look at these results just within a few days, a few early cycles, we can see that this green cell that rolled over very well before the black has much lower coulombic efficiency. These are triplicate cells. This is very repeatable. And really, if we want to understand where this is coming from, we can start splitting up coulombic efficiency into those two terms, that fade-based term, the slippage-based term. And as we would assume, based on what we saw in our long-term cycling, the fade is nearly identical within these short-term experiments. But this slippage-based term is twice as big. The rate of relative reactions happening of these slippage-based mechanisms, electrolyte oxidation, transition metal dissolution, is twice as much in that other cathode. And even though we cycled these 25 times, we could see this just within a few cycles. The trick with this type of cycling, this method here, is we cycle these cells very slowly because we're looking at electrochemical contributions to failure, not necessarily kinetic. So these cells are cycled at C over 10. And this kind of removes any of these kinetic failure mechanisms from the picture. And we're really looking at what's the electrochemical difference fundamentally between these two cells that we've really controlled all these variables. So we use this all the time, and this allows us to not have to chew up hundreds of channels with cells cycling for thousands of cycles. And we can just cycle them for a few days. So another case study, we just talked about cycling cells really slowly, removing kinetics from the equation. And that's where a lot of the papers on UHPC focus on is cycling cells slowly. And a lot of customers ask, do I have to cycle cells slowly? What if I want to see cells failing due to kinetic failure mechanisms? So this was inspired by some work done in the Don lab where they were looking at electrolyte pumping, and Jeff gave a nice talk where he mentioned this phenomena the other day. And what they realized is that there was a difference between what happens when you cycle cells oriented vertically versus horizontally. And this could be related to how the electrolyte moves in and out of the cell when the cell is breathing, say, during charge and discharge. So what we wanted to figure out is, can we use UHPC to detect that this is happening? And how quickly can we do that? So this is just cycling data that was taken on anywhere for 1,000 cycles. So what we did is we had some cells cycling at C over 3 charge, C over 3 discharge, in a vertical and horizontal orientation. And there was a little bit of a difference, but not too much. But it really took hundreds of cycles to start happening. And then we also took some cells and did a C over 5 charge, a little slower, but the discharge was a lot faster in 1C. So this is discharge as the materials relax within the cell, electrolyte comes back into the jelly roll. There's not as much time for the electrolyte to get in, equilibrate, and the concentration gradients to equilibrate within the cell. And what we see is the cells positioned vertically perform a lot better, and the cells horizontally do a lot worse. And again, this takes 300 cycles or so to start seeing this difference. So we said, what if we do this exact same thing? We're not going to do slow cycling on UHPC. Let's just do the exact same protocol on UHPC. And I'm just going to show coulombic efficiency and the charge endpoint slippage because we know we're not going to see much difference in the capacity fade. So for the C/3 cells, both vertical and horizontal show nearly identical coulombic efficiency. This is only 75 cycles too at these rates. So this is not very long-term testing. And the charge endpoint capacity slippage is similar as well. This makes sense because the CE is more or less the same. However, when we move to this faster discharge, all of a sudden we see this really depressed coulombic efficiency and a little bit inconsistent in the cells that are oriented horizontally. And I say significantly different. This plot is really far zoomed in, as you can tell. But for UHPC, this is pretty significant. And this is all driven by charge endpoint capacity slippage. So we're driving some kind of slippage-based mechanisms here, oxidation or something. And there's some a really neat paper published that really talks about this electrolyte movement, concentration gradients that can really drive failure. You can have preferential oxidation of electrolyte localized within the cell. And we're starting to actually be able to measure that very quickly. You can see within a day or so of cycling, just a few cycles, we immediately see this difference that took a few hundred to see earlier. Okay, so another case study I have here is lifetime prediction. So, we've got all these new metrics. We've got these high precision coulombic efficiency. We've got this endpoint slippage and fade that we can now kind of separate out and look at, these degradation mechanisms and separate them, isolate them. Can we use that to now predict how quickly a cell will die, even thousands of cycles out? I showed we can rank things and say something's going to happen first, but can we do this quantitatively? So we partnered with a company called Sandbox AQ, spin out of Google, and they're a machine learning AI company. And so we really focused on our UHPC hardware, our degradation expertise, and then leverage Sandbox for their data pipelines, machine learning expertise, and really just curiosity. How can we apply UHPC data to predict lifetime? So we put together this whole series of cells a couple years prior. Fortunately, we were a bit ahead of the game here and had some cells that had been cycling for a couple years already. And so these were some commercial cells with and without silicon, all with high nickel anodes. And we put them on in this matrix here where we were trying to turn on and off different degradation mechanisms to really challenge the ability to predict lifetime. Not trying to make it really easy to do and only playing with, say, temperature. So we did 3 different temperatures, 25, 40, and 60. We had those 3 types of cells, and then we also played with the SOC ranges. So using the whole SOC range, 2.5 to 4.2 volts, cutting out some of the high nickel cathode upper cutoff or upper capacity, so stopping at 4.06 volts, and then also not utilizing all of the silicon capacity and going only to 3.3 volts to 4.2 volts. So we're really turning some of these things on and off, tuning them so that the cells aren't going to fail necessarily the same way every time. So this makes it really hard to build a generalizable model just using traditional cycling data, just looking at, say, the first 100 cycles of these data. So using some standard modeling features, primarily from the Severson work, that's really famous in the field. So looking at delta QV voltage features and changes in the first 100 cycles, and then the discharge capacity trend in the first 100 cycles. This does a very poor job at estimating the cycle life using these models. However, if we just layer in these UHPC features of these slower cycles, C over 10 cycles, we have our coulombic efficiency, we have charge endpoint slippage, we have our delta V, we have our fade. And we layer some of these metrics in. All of a sudden, we take some of the kinetic information from the first 100 cycles, we layer it in with this electrochemical information about what's happening, and our models perform incredibly well. And so this work is still ongoing with Sandbox. I think I don't have another slide there, but we're looking at ways of using the raw data and DQDV, DVDQ metrics, because what I don't talk a lot about here is how good and useful the raw data is, raw voltage traces of UHPC. Looking at that to inform some of these models as well and create even more generalizable machine learning models. We also have another data set of about 4,000 tests rather than just a couple hundred that we're also working on them with. So just going to go back to this slide from earlier. UHPC, what we're really trying to do with it is show people that you should use it for small things with big implications. Don't expect to buy it and just cycle a cell for 1,000 cycles. Really dig into the data, find these gaps in these use cases that'll really bring value to your development cycles. You don't need thousands of these channels. I've only got a couple hundred, and I've got 3,000 other channels in the lab. So it's just a few niche test cases to really advance your development. Happy to chat about how to help with that, how we can help, how the equipment can help. And thank you.